Greetings to everyone. I hope you are safe, healthy, and warm. My name is Ilias Marmouzi, and I am a postdoc at OSU Center for Health Sciences. I am part of the Neuroinflammation Lab and the Pharmacology Physiology Department. Our team aims to identify new non-opioid drugs that target chronic pain and psychiatric disorders. One of the promising candidates for this multi-target strategy are nicotinic acetylcholine receptors. Nicotinic acetylcholine receptors are expressed in glial cells and involved in the regulation of inflammatory pathways. Based on Dr. Ilias' previous work, we will investigate PAM2, a positive allosteric modulator of the alpha-7 nicotinic acetylcholine receptors. This compound demonstrated previously anti-neuropathic pain effects in mice. In the current work, we have tested PAM2 and nicotine alone or in combination in non-stimulated and inactivated glial cells. To activate both microglia and astrocytes, appropriate doses were selected based on a preliminary study. In the actual experiment, the levels of pro-inflammatory mediators in the culture media were measured by ELISA after interleukin-1 beta activation. As shown here by the percentage of untreated activated control, nicotine at 5 micromolar and PAM2 at 0.5 and 2.5 micromolars all inhibited the release of CXCL10 in activated microglia cells. This effect was potentiated when both of them are combined. Interestingly, methylcaconitin, or MLA, at 1 micromolar abolished this effect, supporting the alpha-7 nicotinic acetylcholine receptors mechanism. More pronounced effect was observed on interleukin-6 and also on CCL2 release. Similarly, astrocytes were downregulated by PAM2, nicotine, and their combination, an effect that was antagonized by the MLA. The most pronounced effect was observed in interleukin-6 release. PAM2 seems to modulate interleukin-1 beta-induced chemokine cytokine production in human microglial and astrocyte cells. PAM2 seems also to potentiate the inhibitory effects of nicotine on interleukin-1 beta inflammation. PAM2 and PAMs in general should be pursued as potential therapeutic agents to treat neuroinflammatory pain. In our future work, we will further investigate the modulatory effect of PAM2 on inflammatory signaling in astrocytes and microglia and elucidate the inhibitory mechanisms related to the alpha-7 nicotinic acetylcholine receptors anti-inflammatory effect. This work was partially funded by a pilot seed grant and by the Department of Pharmacology and Physiology. Please do not hesitate to reach out with any question at the email shown here in this slide. Thank you very much for your attention.